Okay, we're going to do a little activity and discussion here. If you would, get out a piece of paper. I'm going to have you writing some stuff down, but this isn't anything that you're going to be turning in, but it will help if you are writing stuff out as we go along. This is one of these things that we're just doing for learning purposes, okay? Just to help you better understand how the scientific method works. So, the following thing that we're going to look at is something called spontaneous generation, and this was the idea from a long time ago that living things just suddenly arise from something that was non-living. Now we know in today's world we don't believe that, but for a long time, in the mid-19th century, this was an acceptable explanation for some types of life and the rising of some types of life. So what we're going to do is go through some experiments performed by some scientists that were set at basically disproving this idea of spontaneous generation. And the first guy we're going to look at is from the 1600s. His name is Francesco Redi. And he proposed a whole different idea or hypothesis to explain that specific example of maggots that appeared on spoiled food. According to the spontaneous generation theory, rotting meat would simply turn into maggots, and then those maggots would grow or develop into flies. Okay, so we have this dead meat, this non-living material turning into something that was living. Francisco Redi disagreed. Okay, and he made some observations. He observed that maggots appeared on meat a few days after flies had been seen on the food. He then inferred that the flies had left behind eggs too small to be seen, and it was these eggs that were turning into the maggots that would then become new flies. Okay, what we're going to do is go through his experiment here, and then I'm going to ask you to draw a conclusion. Okay, I'm going to draw a conclusion. So we'll talk a little bit here about his experiment. Okay, so I'm just going to scroll down here so I can do a little bit of typing. Okay, and I'm going to highlight here and do some underlining. I think I'll go red here. But some things we need to talk about. We already know what the hypothesis was, is that the hypothesis, let's change that to Redi's hypothesis. Oh, need my red. Was simply that maggots came from eggs laid by flies on the meat. Okay, that's his hypothesis. Okay, so we go from there, and I'm just going to hop down here real quick. Actually, I don't think I want to underline this, so I'm going to get rid of the underline. Let's talk about his experiment. Now, in his experiment here, as we said, we want to use a controlled experiment, and Redi did just that. He has two groups here. Remember, we talked about the control group and the experimental group. So first thing we have to look at are, what are the variables? Okay, what is the variable that he is testing? So, I'm going to tab over, not letting me tab. Let's look at the independent variable. What is his independent variable? Take a second, look this over. Okay, now what do you think the variable is he's testing? He goes to hypothesis, he says maggots came from eggs laid by flies on the meat. Okay, so his independent variable. What is one set of jars getting that the other jars are not? Well, the jars on the left are getting flies, while the jars on the right are not. Okay, and that leads us to our dependent variable. And what is he observing? What is he observing when he's doing this experiment? Take a second and figure out what he's observing. What effect do the flies have on the one on the left? His dependent variable, excuse me here, can't spell, is the presence of maggots. Did maggots show up or not? Okay, so with that said, we have our control group, and that is the jars with covers. And then we have our experimental group, jars without covers. So now what you're going to need to do, and you can stop the video here at this point, is what I want you to do though is take a second and think about the results. So if you want to write down what are the results, okay, and then draw a conclusion. So stop your video, write that down, and then come up with a conclusion, and when you're ready, start the video over again. Okay, so what were the results? Well, the jars with covers allowed, well, I shouldn't say allowed, had maggots on the meat, while the jars should say without here, with covers did not have maggots on the meat. So those were your results. So what conclusion can we draw from that? Well, what did you come up with? Did your conclusion match this? Was Reedy's hypothesis correct? Well, it looks to me like it is. So can we conclude flies must come in contact with the meat in order for maggots to appear? And we could even take this a step further and say the maggots must be coming from the flies. Okay, a perfect example of Reedy using the scientific method to disprove an existing hypothesis. Okay, we're going to move on to another scientist. His name is Lazzaro Spallanzani. And in the 1700s, he designed a controlled experiment to show that life did not arise spontaneously from food. In other words, he was trying to support the work of Reedy. He inferred that some food spoiled because of growing populations of microorganisms. Okay, so microorganisms. So where do these microorganisms come from? Did they just come from the existing food, or did they come from somewhere else? And you can look here as an experiment. We have two um, different flasks. Both of the flasks get heated. Okay, the purpose of heating here, boiling it, is to kill off any existing microorganisms. This one on the left, he leaves open. This one that he on the right, he seals up. He melts down the opening and seals it up, and then he observes the two flasks. He notices that this flask that was left open gets filled with microorganisms, while the one that's sealed does not. Okay, so here's what I want you to do. I want you just to look at these pictures and look over this controlled experiment, and I want you to simply identify the independent variable, the dependent variable, and then identify three variables that would need to be controlled that might also affect the outcome of the experiment. So we're just kind of looking into controlled experiments and the different types of variables here. So stop the video, and when you're ready, you can restart it and see how you did. Okay, let's see how you did here with your independent, dependent variables and variables that needed to be controlled. Now, again, if we go to a controlled experiment, the independent variable is the one that is being manipulated. So in other words, what is getting into one flask that is not getting into the other flask? Okay, and the correct answer here is the air in the atmosphere. When he sealed that second flask, air could no longer get into it. Okay, that might been a little difficult for you, but it's just all you're doing is you're looking at the two flasks and what is one getting that the other is not. Okay, your dependent variable is what are you observing? What gets affected in that? So what did the flask on the left get that the sealed flask on the right did not get? And again, we're looking at the presence of microorganisms. Okay, so that is what we're studying the growth of. So what are some other variables that could affect the outcome here that we should have paid attention to? So we'll just go down through here and list some things. Okay, one of the things that we did to both flasks is we boiled them. Now, something that you would want to pay attention to as a scientist is make sure they were boiled at the same temperature, heated at the same temperature, because if one was hot at a higher temperature and lower temperature, maybe the one with the lower temperature didn't kill off all the already existing microorganisms. Okay, the glassware might be something that you want to pay attention to. Had the glassware been sterilized? Was it the same type of glassware? The size of the glassware? You want everything about the glassware to be the same. Another factor that I came up with was time. Did you boil them for the same amount of time? If you boiled one longer, it was more likely to kill off the microorganisms. Maybe you heated the other one not long enough. Okay, you want to heat them or boil them the same amount of time at the same temperatures and the same type of glassware. Okay, you may have thought of some others, I don't know, but these were the three that were seemed to be most important that I was studying. Okay, so again, if we were to draw a conclusion here, so I'm just going to type down here, conclusion. What would that be? Now, this is the conclusion I was able to draw. You can read through it. Microorganism open flask on the left came from microorganisms already existing in the air that entered. No microorganisms were present in the sealed glass because no air could get in. Therefore, microorganisms found in the broth did not spontaneously occur. Just another example of how scientific method can be used or was used to disprove spontaneous generation I hope this better allows you to understand the importance of the scientific method and how it's used.